Um,
Let me just introduce the debaters and contestants here. This is the second debate in the COVID Origins debate series. Again, we will have two 90-minute presentations, first by Yuri and then by Peter, and then we'll have 45 minutes for each side for rebuttals. So, Yuri, please introduce yourself, and then you're the first speaker for a 90-minute presentation.
sure, yeah, so I'm Yuri Deigin. I'm a biotech entrepreneur and a drug developer, so I'm working on gene therapies for Alzheimer's and other age-related diseases. And I'll be presenting on the lab leak side of the debate, and I've been kind of digging into COVID origins for many years now. So I guess that's the quick introduction.
Also, I'll start your time now.
Oh, so okay, I'll go into the presentation. All right, let me just set my timer. Okay, let's do it. So yeah, I guess I already went with the introduction. I'll be centering on genetics evidence and yeah, from the lab leak side. And well, I guess I already gave the introduction. Just one point to make is that I'll be presenting just the genetic evidence, but I won't be doing the probabilistic stuff, like probabilistic inference of the analysis. I'll leave it up to Sarsh, and he'll do it in session 3. So let's just dive into the genetic aspects of SARS-2, which I think provide compelling evidence for the lab leak hypothesis, because basically SARS-2 is precisely the virus that you'd expect to have leaked from the Wuhan Institute of Virology in 2019, because the characteristics of the virus align with the type of research that WIV was doing at the time. And so it has the virus has several features that are unique or very rare in nature, making it kind of less likely they would have evolved spontaneously in the wild, but more likely that because they're consistent with the stuff you might anticipate from lab-based modifications or experiments, especially in light of the DEFUSE proposal. And also, in addition, the low genetic variability of the virus early on in the pandemic is very surprising, because in nature, viruses tend to have higher degree of variability as they evolve and adapt to hosts, but the kind of limited variability of SARS-2 suggests a quick localized jump that was of a virus that was already well suited to humans. So, and that's the kind of behavior you'd expect if the virus was like pre-selected for human infection, and maybe even further adapted for human infection in the lab, in like humanized mice or human cells, etc. So let's go into point by point into basically the first section of that I outlined in the previous slide. Basically, the main point is, or the first, I guess, the first major point is that SARS-2 is just very similar to what the Wuhan Institute of Virology was already working on and was planning to work on in 2018. And in light of that, as I said, SARS-2 is precisely the kind of virus you'd expect to have originated from the Institute in 2019. And in the next two slides, I'll just go into specific genetic features of the virus that make that connection highly plausible. But before that, what did the Wuhan Institute of Virology work on? Well, they've been the lab most actively involved in collecting and researching various coronaviruses, particularly those that are similar to SARS and MERS, basically the viruses posing the biggest threat to humans. And before the pandemic, they boasted of having a collection of like many viruses. Like they had an internal database of like 20,000 samples, and that database mysteriously went offline after the outbreak. And so they were like the lab doing this kind of research. And of course, they didn't just go out to these remote caves to collect these viruses. They also genetically modified them in what's called gain-of-function studies, which basically means manipulating viruses to understand their potential for infecting humans and like pathogenesis in humans, but also in the process that can make those viruses more potent or more adapted to humans. And of course, this poses a huge risk of pandemics, because if those viruses escape, they're already like well adapted to humans and easily transmissible. And last but not least, an important area of interest for WIV was furin cleavage sites, and that's important because adding a novel furin cleavage site can greatly expand viral tropism. For example, if you add a furin cleavage site to a bat gastrointestinal virus, that can turn it into a human respiratory virus. And so this presence of an FCS in SARS-2 is particularly interesting because, well, like I said, it greatly it changes a bad gastrointestinal virus into a human respiratory virus.
Yuri, since we're going to be talking about furin cleavage sites a lot, could you perhaps just give a quick definition of it for the audience?
Sure. I wasn't planning on it, but...
Sorry to interrupt.
Furin cleavage site is basically a protease cleavage site in a coronavirus that makes it cleaved by furin. Furin is one of the proteases that cleaves particular recognition sites that are recognized by this enzyme, and this enzyme is generally expressed in airway epithelial cells, both in humans and other animals. And so, of course, the virus must be cleaved before it can actually enter cells. So it can cleave by different enzymes, but if it can be cleaved by furin, that makes that virus much more potent, much more tropic to certain cell types that express the furin enzyme. Kind of back on the presentation, furin cleavage sites were a very kind of popular topic in coronavirology in 2019 and in 2018, because in 2018, of course, WIV and Ralph Baric, they co-authored the DEFUSE proposal, which talked about engineering novel furin cleavage sites, and other groups seemed interested as well. A case in point is the 2019 paper from Beijing, which described engineering a novel RRKR, furin cleavage site in the chicken coronavirus. But maybe more intriguing is Ralph Baric's presentation in China that very same year in 2019. And of course, Ralph Baric is probably the most renowned coronavirologist. And so he discussed the creation of novel chimeric coronaviruses, and he wasn't referring just to the receptor binding domain of the spike protein, but in his remarks, he was hinting at broader modifications that incorporate changes to other virulence determinants or modulate host pathogen interactions. So basically, given his prior collaboration with WIV in 2018 on the DEFUSE proposal, and given his subsequent paper from his team on MERS-like virus cleavage sites in HKU5, I think it's clear that Ralph was pointing to modifications to spike protein cleavage. And it's interesting that while Baric and Zhijiang Li of the Wuhan Institute of Virology, while they collaborated in 2015 to introduce a furin cleavage site into a HKU4 virus, a MERS-like virus, this 2019 paper by Baric on HKU5 cleavage site was not in collaboration with the Wuhan Institute of Virology, despite their just co-authoring DEFUSE just a year earlier. So it's possible that by this time they were actually competing. By 2019, they were actually competing, like Baric's lab and Zhijiang Li's lab or WIV. They were competing to uncover the potential of furin cleavage sites in novel coronaviruses. So now let me talk about the crown jewel of the lab leak hypothesis, the DEFUSE proposal. So in March 2018, this proposal titled Project DEFUSE, Diffusing the Threat of Bad-Borne Coronaviruses, it was submitted to DARPA by EcoHealth and WIV, and jointly they were seeking $14 million for a huge research program whose goal was to collect a whole bunch of viral samples from bat caves in Yunnan in order to find strains that could potentially trigger a pandemic. And then, this is funny, they were actually planning to go out to remote bat caves and immunize bats against this very same viruses. So maybe they should have sprayed them with ivermectin. I'm sure Brett Weinstein would support that idea. And my slides are getting a little out of alignment because of this app that doesn't allow me to share the screen. Anyways, basically, the first phase of the DEFUSE proposal was first identifying these dangerous viruses and that pose a threat to humans before actually diffusing them. And they were this particular proposal was focusing just on SARS-like viruses, the same family that SARS-1 and now SARS-2 comes from. And it's interesting that either the DEFUSE authors had this incredible foresight about a lurking SARS-2 progenitor that was only missing a furin cleavage site that was waiting to spark a global pandemic, or the other alternative being that DEFUSE was actually a self-fulfilling prophecy. This is why DEFUSE is so relevant to our discussion, because it outlined not just sampling many novel SARS-like viruses, but also genetically modifying them to model if they could evolve to become more dangerous to humans. And so in particular, they talked about, first of all, looking for furin cleavage sites in these viruses, and then if they didn't find them, basically creating novel furin cleavage sites in SARS-like viruses. And so in the next few slides, just wanted to highlight some key points of that proposal that I thought were particularly relevant. And so, for example, here, just wanted to highlight that they were talking about sequencing full-length genomes of novel viruses, and then also synthesizing them like into live viruses, essentially creating novel backbones and turning them into live viruses. This is what they mean by like recovery recombinant viruses. And I mean, they had plenty of viruses to study. DEFUSE itself mentioned that they already had 180 sequenced but unpublished SARS-like viruses, and then they talked about including into the analysis of DEFUSE 10,000 samples from like 6 Southeast Asian countries, and they planned to do a lot of live virus work at the WIV. And also I wanted to point out that for the screening of the viruses that pose the threat to humans, they plan to look not just at genomic data, but also at computer binding modeling, and also looking at like 3D protein structure of the spike trimer. And so some critics of the lab leak hypothesis say that SARS-2 would have been like uninteresting because it's only 80% close to SARS-1. But of course, like in the DEFUSE proposal, SHCO14 is like 88% close to the spike. But like besides all that percentages, if you actually do protein modeling, you'll see that the SARS-1 and SARS-2 spikes are actually very close in the protein. And so sometimes you can have like different amino acids in the protein, so like they wouldn't be identical on the protein level, but functionally you can have similar amino acids, and functionally the protein can actually be quite close. And of course we know that SARS-2 is very good at binding the human ACE2 receptor, and even better than SARS-1. So if they did some kind of modeling on the spike, I think that would have been apparent just how potentially dangerous SARS-2 is, or the spike in SARS-2 kind of viruses to humans. Okay, also I want to point out that while they were planning to use chimeric viruses for their initial analysis, they also plan to then validate any interesting results from that analysis with full-length viable viruses, like essentially creating novel backbones, as I already pointed out in the previous slide. And as I said, the live virus work was planned to be done in Wuhan, in the Wuhan Institute of Virology. And I think this is probably the most important page in all of DEFUSE, as it talks about the very same sort of like gain of function genetic engineering that they plan to do that I think could lead to SARS-2. So here, like they say, we'll synthesize quasi-species with novel combinations of mutations to determine the effects on specific genetic traits on spillover potential of future and unknown recombinants. And the most striking part here is where they propose engineering novel furin cleavage sites, and then testing how these modified viruses would grow in the human airway epithelial cell cultures. And also then seeing how pathogenic they could be to humanized mice, basically mice tailored to have human ACE2 receptors in their lungs. And, don't want to like belabor the point, but SARS-2 is unique in its family of like 1,500 known SARS-like viruses, unique in having the furin cleavage site. None of the other 1,500 SARS-like viruses have a furin cleavage site. And so in light of like the intentions outlined in the DEFUSE proposal, it's very, very suspicious. Oh, and sometimes zoonosis proponents dismiss the DEFUSE suggestion of engineering novel furin cleavage site because they say that was only meant to be done in chimeric viruses. I'm not sure why, but in any case, DEFUSE in a couple of places, like this is the second time I'm having basically pointing this out, that they plan to validate the results from chimeric viruses with in full-length genomes. So basically, they were anyways planning to create novel backbones. They were saying like we will recover 3 to 5 full-length novel viruses per year, but also if chimeric viruses showed some interesting results, then would lead to them validating those results in the full-length viruses from where the spikes for those chimeras came from. And they would then look for viral growth in primary human airway epithelial cultures and in vivo, and basically in these cell lines, the furin cleavage site, if they inserted it, it would not mutate away. Because in some cell types, like Vero cells, for example, the furin cleavage sites tends to mutate away because Vero cells either don't express furin, they have very low furin expression, so there's like selective pressure against the furin cleavage site. but in human airway epithelial cells, the furin cleavage site would actually remain. And so basically, when someone sees the DEFUSE proposal, I think it becomes very suspicious that the unique features of SARS-2 align so well with what the DEFUSE proposal was talking about. And I mean, this table just highlights some of these key features, like the ACE2 receptor binding. Of course, they were talking about this being the primary screening factor for picking which viruses to work on. And SARS-2 is obviously very good at human ACE2 receptor binding, maybe even like human receptor is the top match out of all known species for SARS-2 receptor binding domain. And second, they talked about N-Glycan manipulations, and I'll talk about this in detail. And SARS-2 has a missing N-Glycan, and this feature actually greatly increases infectivity for human cell lines. So this is also quite interesting. And finally, furin cleavage sites. That's I think I already talked a lot about this, but it's just very, very weird that SARS-2 has that unique feature. But just to make sure, like, we're not cherry picking, we've put together this table of everything that DEFUSE talked about, and also whether we see evidence of that or not in SARS-2. So just to quickly sum up what DEFUSE was about, it was concerned with novel SARS-like viruses that pose a higher risk of spillover to humans, so they were going to screen spikes from a whole bunch of SARS-like viruses, and then if they saw good binding, then they would also verify this in vitro by slicing those spikes into chimeras, and if they saw again good results from chimeras, they would validate those results either in culture or maybe in live humanized mice. And of course, the goal was to see also the pathogenicity that these viruses pose to humans, and humanized mice are a good model for this. And
Yuri, I wanted to ask a question about this slide as I was reviewing, and just a kind of a higher level question. A lot of the proposed work in DEFUSE, and much of it you've highlighted here, seems like it would leave vendor records, something where either they've purchased H2 mice or they talk about primer walking, they've got to purchase all those primers. There are all sorts of things that might give evidence to which of the DEFUSE activities actually happened, since it wasn't funded. Has anyone looked for that information? Would it all be Chinese vendors and extremely hard to come by? Would any of it be from Thermo, IDT, etc.? Can you speculate, comment?
Actually, SARS said questions will be at the end, and then this kind of gets. First of all, I didn't plan on answering them now because that kind of takes away from the time, and I'm borderline on 90 minutes. So maybe we can.
Okay, we can address it later, but we had planned. I was under the understanding we had planned to ask questions during presentations.
Well, yeah, that way I won't make it 90 minutes. Sorry,
we can save on the interruptions, I think.
Okay, we'll save. I'll save that till later then.
Okay, yeah, I can definitely address those questions later on. Okay, so back to the slide. It just summarizes that what the DEFUSE plan to do and how we might see some of these things manifest in SARS-2, and of course, DEFUSE was not WIV didn't have to follow DEFUSE to the letter, because DEFUSE was not gospel. They could do whatever research they wanted, and DEFUSE was essentially just the minimal, an abstract of a research paper, and it had one-line things about we'll generate novel furin cleavage sites. But of course, once you actually dive into the research that you plan to do, it can be much greatly wider than what you originally planned or what you described in the research proposal. And of course, also in terms of funding, just because DEFUSE wasn't funded, obviously WIV didn't need the funding to do the things that they were interested in. They had plenty of Chinese funding. They were doing this type of work anyways from Chinese funding for years. They had all of the viruses in their collection from decade plus of collection of sampling bats, and in terms of vendor records, just like it's hard because that thought is already in my head. I'm thinking about it. I'm not sure what kind of vendor records they would need if they were synthesizing the backbones of the virus. They would probably do that in house. So because they created backbones of the viruses in house for the PhD thesis. Like they had 2 different backbones for WIV1, and so even if they used outside vendors, they probably would be Chinese vendors. So anyways, kind of going back to the DEFUSE proposal, as I said, there's some interesting aspects of DEFUSE that, or interesting aspects of SARS-2 that sync well with DEFUSE. Of course, the primary one being or one of them being how well it binds to the human ACE2 receptor, which of course was the selection criteria for them to look at interesting viruses. And of course, several computer models and infectivity experiments have shown that the human receptor is like, if not the highest, among the top highest species in terms of binding affinity of the SARS-2 receptor binding domain. But one thing I want to point out is that just because, that alone, the high affinity to the receptor, that probably would not be enough to spark a pandemic, because you need a furin cleavage site, essentially. Because we have experimental results in non-human primates that show that without a furin cleavage site, this virus remains an enteric gastrointestinal virus rather than a respiratory virus. Because basically we have, like in the wild, we have viruses like Banal52, Banal236, and they have the same receptor binding domain as SARS-2, but they don't have a furin cleavage site. And so there's been experiments by Marc Eloise's group at the Institut Pasteur, who actually, he's the one who discovered Banal52 and other Banal viruses in Laos. And so they've done experiments that showed that those viruses remain enteric. Those viruses without a furin cleavage site remain enteric, while in non-human primates, in macaques, while SARS-2 actually becomes a respiratory virus in these animals. And this was actually corroborated by observations of guano collectors, people who collect bat guano in these caves where these viruses are actually present, like Banal52. And those guano collectors, they didn't have antibodies to like SARS-2 or Banal52, basically meaning that they were not infected by these viruses. And so I think this kind of syncs up well with the DEFUSE proposal, which was interested not just in the affinity of the receptor binding domain to the receptor, but also in the role of proteases, and especially like human-specific proteases, in like unlocking the barrier to zoonotic emergence, as like one other paper said, that protease cleavage unlocks the barrier to human emergence. I think in the 2015 paper with Ralph Barrick, where they engineered a null furin cleavage site in a MERS-like virus, that HKU4 that I mentioned before. So again, that's out of alignment. Besides the furin cleavage site, which I'll do an even deeper dive later, there's another unique feature of SARS-2 that corresponds to something that DEFUSE talked about. And this feature is a missing N-linked glycan at position N370. And this ablation of the glycan has not been observed in any natural relative of SARS-2 to date. And what makes this particularly noteworthy is that the ablation of this glycan significantly enhances the infectivity of SARS-2 in human lung cells. But at the same time, this mutation seems detrimental to ancestral bat viruses, because in the bat stomach, it's actually detrimental. And this is actually similar to furin cleavage sites, which are also potentially detrimental for enteric transmission, and so for which there's also selective pressure against in bats. And so DEFUSE proposal's interest in N-linked glycans, I think, well, it can be traced to the observations about SARS-1, that it lost 2 glycans in civet strains, and they speculated that this potentially helped it jump into civets, and then of course from civets it jumped into humans. And so they were interested in a role that glycans can play in like host switching mechanisms. And so this is the direct quote from the DEFUSE proposal. Basically, it's talking about like what I just said, that the ablation of these two N-link glycans played some sort of role in the virus's first jump into civets or raccoon dogs or both. Well, civets are the strains that we actually have in which those glycans are missing. At the same time, the DEFUSE authors now knew that in the original bat strains, the glycans were still present, so they went missing somehow, and then they infected civets, but then they reappeared during the human transmission, and so basically the DEFUSE authors were wondering what kind of role this ablation of these glycans might have played in helping these viruses jump into civets. And so they're citing this 2007 paper, the DEFUSE proposal, basically in connection to these two N-link glycans, and they're showing that basically these two glycans at positions 227 and 699 are missing in earliest civet strains like this one and this one, but then they gradually reacquire them and preserve them in human strains. And so this is one of the motivations they list in the DEFUSE proposal for why they wanted to look at N-link glycans, and another motivation was the role that the glycans play in this DC sign binding, this binding to alternative receptors. And also, of course, as I mentioned, they said that while those glycans were missing in civet strains, they weren't missing in progenitor bat strains, so they had this kind of flip-flopping role between bats, civets, and humans, and so they wanted to investigate that. But in terms of the DC sign binding connection, they then cited a 2006 paper that talked about this particular glycan that they identified in SARS-1 that they think played a role in DC sign binding. And it's interesting that the very same glycan that they mentioned in that paper is the same glycan in SARS-2. Like N370 in SARS-2 is the same as N357 in SARS-1. That was linked to this DC sign binding. And so, of course, N370 is not the same glycan as the civet glycan, but it's the same glycan as this DC sign glycan. And that section in DEFUSE, it talked about glycans in connection to DC sign binding, which is a receptor in macrophages. And basically the idea is that maybe if you ablate this glycan, you can increase the virus's ability to infect other tissues. Maybe you have a reduced titer in macrophages, but maybe it now has the ability to infect other tissues because macrophages play a role in immune response to it. But I won't speculate like exactly why they were interested in this. And also, like, I'm not saying DEFUSE is the exact blueprint that WIV would have followed to the letter. Again, like they didn't have to ablate the very same glycans, but it's just showing that they were looking at glycans. And if someone later, inspired by DEFUSE or just in general interested in the role that glycans play in host switching, and someone had this panel of various viruses that they wanted to see the role of glycans, they could have been ablating them one by one and seeing how that affects infectivity, and maybe noticing that increases infectivity in human cells, human lung cells. And then they would have done, for example, in the SARS-2 progenitor, this very same experiment that would have resulted in the loss of this glycan. And yeah, basically it's the importance of this glycan in SARS-2 is that it greatly increases infectivity to human cells. And this is a quote from a study they did, basically showing that when you have this mutation, it increases infectivity in human cells by like 60 times. Basically, if you restore the glycan, the mutant, the wild-type mutant, it has a 60-fold lower infectivity than the current SARS-2 that has the missing glycan. And just to provide some comparison, like the D614G mutation, the early mutation that is known to have increased SARS-2 infectivity, it only boosted it by like 3 times. So that's a huge, then this glycan plays a huge role in increasing infectivity in human cells. And so I think again, in light of the DEFUSE proposals, focus on N-Glycans and their potential role in host jumping, and as we now know, infectivity. I think it's very, very interesting to see that SARS-2 is missing one of the glycans that potentially DEFUSE might have been interested in. And it just keeps getting out of alignment. Okay, and as I said, while the presence of this mutation in SARS-2 significantly increases its infectivity to human airway cells, in bats this mutation is actually detrimental, and so it's not likely to have arisen there. And this is because these viruses predominantly rely on like oral fecal transmission route, and this mutation is, as this quote shows, is detrimental to oral fecal transmission, basically because of the like low pH in bat stomach. So, well, I won't get into the details, but basically they did a study that they restored this mutant and checked how that like what the implications were on the virus, and they showed that the mutants lost almost all of the infectivity under the conditions of a bat stomach, while the wild type viruses, the viruses that actually had the glycan, they retained a significant portion of the infectivity. And so basically this tells us that while these the ablation of these glycans is an advantage for human infection, like in human airway cells, it's actually detrimental to the virus's ability to transmit via the oral fecal route among bats. So this kind of, well, I think it maybe not rules out, but like greatly decreases the probability of this mutation arising in bats. Okay, so like setting DEFUSE aside, even if we didn't know about DEFUSE, like we didn't know about DEFUSE until like summer of 2021. So, but even then, when we see some features of the SARS-2 genome, some of them are like very, very surprising and like not in line with evolutionary expectations. As Christian Andersen said early on, he was the original lab leaker. Of course, the furin cleavage site is probably the most distinguishing such feature. And I mean, of course, sometimes furin cleavage sites do occur in coronaviruses, like as a family. In SARS-like coronaviruses, SARS-2 is unique among like 1,500 known SARS-like coronaviruses in having this furin cleavage site. So this is just so much like unexpected to see a virus from this family with a furin cleavage site, because also, as I said, there's very likely selective pressure against it, because it's actually detrimental for the gastrointestinal tropism of these viruses. Because it shifts, like if you add a furin cleavage site, as Mark Eloise has shown experimentally, that shifts the tropism from enteric to respiratory tropism. And of course, bats prefer to have enteric tropism. And just not only that, but also the way in which the furin cleavage site arose is also very unexpected, that it's not like a single point mutation, but it's which would have been enough. Like there's some viruses which are only like one nucleotide mutation away from having a furin cleavage site arise, but this furin cleavage site was introduced by like a 12 nucleotide insertion at like this very same spot that created the furin cleavage site, which is very suspicious. At the same time, like given that the pandemic, I mean, this was originally a bat virus, so for us for this bat virus to then kind of start infecting humans, it's not unreasonable to anticipate some unique feature that will allow us to do this. So by itself, the furin cleavage site, while it's very kind of unusual to see, from a probabilistic assessment, I think, we're not giving it a very high weight. But the particulars in which it came to be, they are the more suspicious. As I said, the clean insertion, the 12 nucleotide insertion in this spot that created it, as well as also the choice of the codons, the CGG codons for arginine, the CGG doublet. It's a very unusual codon choice for coronaviruses as a whole, and but wouldn't be unusual for genetic engineers. And not only it's unusual, we've looked at for whether there are other SARS-like viruses that have a CGG arginine doublet. And in response to like Tyshkovsky and Punchin, we analyzed 255 existing coronaviruses, SARS-like coronaviruses, and we haven't found a single one that used anywhere in the genome a CGG arginine doublet. And I mean, it's just very, very unusual to see, and also to see an insertion in the genome in general, but also that precise spot of the genome in which it's inserted. If you look at the alignment of SARS-2 relative to its closest ancestor or closest relative, Banal52, you'll see just like how close they are in the latter part of the genome, basically how rare even mutations are. And so it's unprecedented to have such a major like insertion, a pretty long insertion that has such an important functional consequences to occur in this spot of the genome. So basically, this is an alignment of the entire latter third of the virus of the genome, and it's just trying to show like just how rare even non-synonymous substitutions are in this part of the genome. And basically, yeah, it's pretty much the insertion is unique in this part of the genome, this long of an insertion. And that alone is surprising, but of course the functional significance of this insertion being at the very precise spot of the cleavage site is even more surprising. So, I mean, these are just looking at the alignment, and basically you have some hotspots of single nucleotide mutations. Some of them are synonymous, some of them actually change the amino acid, but there's just not too many places where you see such mutations. And then you kind of see this horn of the unicorn, to borrow from Alina, the analogy that just sticks out. And it's not just any odd place in the genome where it occurs, but it occurs precisely at the cleavage junction of the S1 and S2 subunits of the spike protein. And like in the vicinity of this insertion, there's just again very few mutations and very few non-synonymous mutations, and yet it kind of sticks out as being in this very kind of low mutational area, and yet it created this furin cleavage site. And I mean, the later part of the genome, sometimes you have these mutational hotspots, but that was not like that general vicinity of the spike protein was not a mutational hotspot. And I'm out of alignment again. So yeah, it's just very odd to see an insertion in general, because insertions in general are pretty rare beasts, and this slide just try to show how rare they are, because we have a very good now picture of the mutational propensity of SARS-CoV-2, because we now have like 10 million SARS-2 genomes sequenced throughout the pandemic, and after like it infected close to 1 billion people probably by now. I mean, this is like, anyways, so we have this distribution of the mutations, and insertions are by far the rarest mutation. This green line in this bar chart are insertions, and this is the spike, and you see that it's like very, very few, like insertions represent very small percentage of mutations in the spike. And moreover, most insertions are actually short, and 12 nucleotide long insertions are also rare. And of course, insertions really have to be in a multiple of 3 to preserve the reading frame, because codon is 3 nucleotides long. And so we have like all throughout 10 million known SARS-2 genomes, we have like 35 12 nucleotide long insertions after like years of evolution in probably 1 billion people. And yet, of course, most insertions don't even happen in the spike, as shown here. So, not to belabor the point, but it's a very, very unusual insertion to see in a SARS-like virus. And as I said, another interesting aspect of it is that the insertion has arginines coded by the CGG codon, and it's a very rare codon for SARS-like viruses, and in general in viruses they don't like this codon. And as I said, we looked at not just the codon but at the doublet, the CGG doublet in our paper in response to Tyszkowski and Panchin, and we haven't seen this doublet in any of the other SARS-like virus that we looked at. And basically 2.6% is the frequency of arginines being used by, being coded by CGG in the SARS-2 genome. And there was some questioning whether, is it exactly 2.6 or maybe it's a little higher. And we found another source for our numbers, and basically this is a very nice table that shows all of the arginines in each gene of the coronavirus, and then we can just count up which ones are coded by CGG, of course, omitting the insertion. And basically, without the insertion, only 2.6% of arginines in SARS-2 were coded by CGG. And moreover, in the spike gene, only the insertion has CGG. So in the spike, it's even less likely for us to expect to see a CGG codon to be used, let alone two. And of course, you might ask, why would any lab or WIV or whoever use CGG codons if they were to insert a arginine furin cleavage site? And first of all, we don't know the exact motivation of somebody, but there's some plausible explanation. The first one being just it's the most top of mind codon for human genetic engineer, as this table shows from the Moderna and Pfizer vaccines. When they were codon optimizing their spikes for the vaccine, they recoded either almost all of the arginines to CGG, or the arginine starting with the C to CGG. And so it's just I think it's a natural codon for human genetic engineer to use, just because I think it's in the human genome, CGG is actually either the most frequently used codon or like at the top of the codons being used for arginine. Also, this is my favorite part. CGG creates a new restriction enzyme site for this FAO1 restriction enzyme that could be useful in tracking the newly created furin cleavage site, because as we now know, these furin cleavage sites can sometimes get deleted if you passage it in like the wrong cell culture that doesn't express furin or has very low furin expression. And so if you're tracking the evolution of a virus, as if you're a genetic engineer, it's nice to have this easily accessible tracking beacon in the form of a restriction enzyme site that you can quickly do digest and like run a gel and see if your furin cleavage site is still present in your viral colony or if it mutated away. And when I pointed out, Peter mentioned that he hasn't observed anyone in virology using FAO1 before, but it actually has been used. And so here are like examples of a couple of papers, and one of them actually used FAO1 for this R-flip analysis that I mentioned, or hypothesized could be useful for SARS-2. And R-flip stands for restriction fragment length polymorphism, and it can be used to identify the presence or absence of specific restriction enzyme sites, which can then be used to like quick screening of deletions, for example, of the furin cleavage sites. And like I said, because furin cleavage sites, as we now know, are prone to deletions in SARS-2. Okay, now let me address another aspect of the furin cleavage site, the leading proline. Basically, some say RRA would have been enough to create a functional furin cleavage site, so why does SARS-2 have PRRA as its insertion? But even some of the proponents of the zoonotic hypothesis, they've suggested in their studies that RRAR by itself, without the proline, might not be actually all that well cleaved. But if you introduce a proline, it creates a turn in the protein, which actually could expose the S1, S2 junction of the protein, that loop that contains the null furin cleavage site, and that can actually enhance its cleavage. And I mean, yeah, that's just kind of one idea, but just to kind of like dismiss the whole lab leak origin hypothesis by saying like nobody would insert a proline, I think it's not a good argument in general. I mean, genetic engineers could have their like many reasons for why they would want to use to include a proline in their like leading proline in their insertion in their furin cleavage site, because for example MERS, the MERS coronavirus has a leading proline in its furin cleavage site. Some feline coronaviruses do, and WIV was like they studied MERS a lot. In fact, they engineered a novel furin cleavage site in the MERS-like virus, showing how important the furin cleavage site was for unlocking human tropism. They also studied feline coronaviruses, and at the same time, there's some naturally occurring coronaviruses like close to SARS-2 that have very close resemblance to the SARS-2 furin cleavage site. And for example, the viruses like RM102 and Banal 116, they have this PAAR sequence at the S1, S2 junction. Of course, PAAR is not cleaved by furin, but it's because you need arginines. And so basically, if you insert RR in there, and if you turn PAAR into PRRAR, that that actually gets cleaved into furin by furin. And so they could have been modeling the evolution of like what would happen if this PAAR evolves into some version of a furin cleavage site. And they could have had like several candidates for cleavage sites, from like the most minimally cleavable to like very highly cleavable. They could have had a panel of various furin cleavage sites they wanted to evaluate. And it's actually very interesting.
45 minutes.
Because if you it's interesting because if you look at the actual nucleotides of the PAAR fragment in these natural viruses, they use the same codons like CCT for proline and GCA for alanine as is used by the PRRA insertion. And so it's like if someone took the PA fragment and inserted from these natural viruses and just inserted two arginines in between. So, I mean, of course, you could say it's just could also happen just by chance, but it's just a very interesting coincidence in the series of a whole bunch of very interesting coincidences. And so basically, yeah, this is what could have happened is a genetic engineer would find a RM102 like virus, see PAA fragment in there, and decide to model like the evolution of that into PRRAR furin cleavage site. And so I think it's quite plausible that somebody at the Wuhan Institute of Virology or any other lab in Wuhan looking to, inspired by the DEFUSE proposal, looking to carry out this kind of research into modeling the spillover potential of these viruses could end up with a furin cleavage site with the leading proline. Okay, so in summary, the furin cleavage site in SARS-2 stands out not just because of its existence, but also because of the way that it came into existence. And of course, while it's not impossible for a virus to evolve a furin cleavage site naturally, the conditions and characteristics surrounding the SARS-2 furin cleavage site, I think they make it a unique case, especially in light of selective pressure against an FCS in bat SARS-like coronaviruses, and well, in general, it just kind of very unusual to see such a novel feature arise, like a novel trait which was not seen before in any of the member of the family arise in a virus that jumped species. And also, I think we'd expect such a feature to arise via single nucleotide mutations, or just mutations rather than an insertion, because, as I said, some of these natural viruses are just like one or two nucleotide mutations away from.
He's frozen for me.
Like that way.
I can see you. Eric's there.
Site,
yeah, I just lost Yuri.
I think this shows that there's actually selective pressure against furin.
I paused his time. I'm gonna
And also insertions, of course, they can.
This is like the part of the
but
Tour de
they're
France
typically
when the rider
smaller,
falls down and I have to
the
go
long
back
ones
and
usually come
help.
from the same virus. This is another aspect that, we still don't know where the insertion came from. Like, what's the donor sequence for the insertion? And usually the donors come from the
Yuri,
same.
hang on.
Sorry.
Yuri, you cut out for about 15 seconds, so I'm not sure what we missed.
More than 15 seconds. None of us could hear you.
Yeah. So I paused your time. I don't know how far back you want to go. Maybe 30 seconds.
The start of this slide is probably good.
Okay, start of the slide. Yeah. Basically, I was saying that it's interesting that it's not just the way that the kind of key observation is not just that we have this furin cleavage site in SARS-2, but also the way it came into existence in SARS-2 that is very suspicious. And basically, also I think I was saying that the way that it evolved using an insertion is very suspicious or very unusual, because usually like new traits arise via mutations, and this trait could have easily arisen via single point mutations, because viruses like RM102 or Banal 116, they're just one mutation away from a furin cleavage site arising, and yet we see this arise via this very uncharacteristic 12 nucleotide insertion. And so insertions, they can happen, but usually they're smaller insertions, and usually we actually know where they came from, like the donor sequence of the insertion. And usually the donor sequence is either the same virus, same genome, because it usually happens during recombination events when the virus replicates. So either the same virus or some sort of like virus in the family that infects the same cell along with this virus. But in this case, we still don't know where the insertion came from. Like, what's the donor sequence with these 12 nucleotides that gave SARS-2 the nucleotides for the furin cleavage site. And so, and finally, the codons, of course, they're also suspicious because they're not used. First of all, viruses like SARS-like viruses don't like these kind of codons, and we don't see them in the spike all that often. So, in light of all of these things, I think it's just SARS-2 is again inconsistent with evolutionary expectations, as Christian Andersen pointed out. Okay, let's move on to our third point. It's just it's intriguing that SARS-1 exhibited very low genetic variability pretty much from the start. And so typically when zoonosis happens, the virus actually has very high mutational rate because it's trying to adapt to a new host. It's really kind of experimenting and trying to see. I won't use like anthropic language, but basically with SARS-1, for example, we saw a very high mutational rate, and it made multiple jumps into species. It jumped into civets several times, and from civets to humans several times, and this is not what we observed with SARS-2. In fact, we see just a single jump and pretty low genetic variability, so it seemed that SARS-2 was either pre-selected for human transmission or even pre-adapted for human transmission, which is, of course, much better explained by a lab leak than by zoonosis. And because under zoonosis, we still don't know how exactly it would have happened. And so basically in this section, I'll show that genetic data from early SARS-2 cases, they show a very low level of genetic variability, which is unusual for zoonosis. And also I'll show that the market was dominated by a later strain of the virus rather than the earlier strain, the earlier lineage. And like if we're to believe that the virus spilled over in the market, this is surprising because we'd expect the earlier lineage to dominate the market, and yet that's not what we observed. And so in totality, these observations, I think they're more compatible with lab leak than zoonosis. Okay, so in terms of the low genetic variability of the early cases, let me just talk about these a little bit. So basically, this is a diagram of 549 of the earliest SARS-2 genomes sampled between December and January, December 2019 and January 2020, basically the very start of the outbreak. And this diagram shows that the early cases split into like two distinct lineages, each kind of radiating from this intermediate genome, AB genome. But what's striking is a small number of mutations that separate the root of the lineage from like the outliers. Basically, it's just either 4 or 5 mutations. And I think that's a very narrow genetic spread, especially considering the number of viruses sampled here. And so such low genetic variability, to me, indicates that SARS-2 was already like pre-selected or pre-adapted when it encountered humans. Also, the prevailing consensus is that lineage B of the virus evolved from lineage A because lineage B is 2 mutations farther away from the bat ancestor. But there's one research group that actually tries to claim that they were 2 separate spillover events. And this research group, led by Picard, proposed that the 2 lineages actually represent 2 separate spillovers from animals. And they base their claim on 2 main points. First of all, they just disregard any A and B intermediates, and like on this slide shows there's some AB intermediates, but for their model they've disregarded them. They're saying like these are all sequencing errors. And second, they developed this Bayesian model to simulate viral evolution based on like sampling dates rather than just phylogeny, because phylogeny is pretty clear that B descended from A, but so there's like no reason to suspect 2 spillovers. But of course with their model, like initially they had pretty considerable strong base factors, but it turns out that they actually had bugs in their code, and after you fix those bugs, that reduces the base factors by like 6 times and pretty much puts them into like very, very weak probabilistic support. Essentially, I think that pretty much dismisses the hypothesis right there. But also, the genetic difference between those 2 lineages is just it's very small, just 2 mutations, and such a slight difference. I think that alone is unusual for 2 spillovers, and also these like 2 mutations can happen between even in a single spillover between like 2 consecutive transmissions. And here we have data from the Diamond Princess, the cruise ship, that showed that in some cases you can have like 2 mutations or even 3 mutations in a single transmission event. So between 2 people, basically one person can evolve 2 or 3 null mutations. And I think this point is critical because it shows that the absence of intermediate genomes is not sufficient to conclude that there were separate introductions. So it's entirely possible that lineage E could have evolved from lineage A through a single transmission.
Yuri, go back 15 seconds. You dropped out again.
You cut out when you were saying that there could be 2 or even 3 mutations in something.
Okay, I was basically showing that in a single transmission between 2 individuals, you can have 2 or even 3 mutations arise in a single person. So the absence of intermediate genomes cannot be used as evidence that there were multiple introductions. It's entirely possible that with a single introduction, you can have someone evolving 2 mutations, like the difference between lineage A and lineage B. And I think Picard are the only ones who are positing 2 introductions, and there are several other models. None of them have 2 introductions. And moreover, as we've shown in our paper, Picard actually threw out many intermediate genomes that we think are valid intermediate genomes that should not have been thrown out. And moreover, after they published the paper, we found several more intermediate genomes from Wuhan itself that weren't included in the Picard model, and I think should have included. But I think all of this is a bit of a moot point, because after the fixing of the bugs, the errata that they actually now published, Picard has published, which reduced their base factors by like 6 times. I think by now this 2 spillover hypothesis is essentially like dead in the water. And also, the earliest sampled lineage A genome already had an extra mutation away from the bad consensus. And this suggests that lineage A was circulating for some time before it was sampled. And so the timeline used by Picard to infer infection dates, because as I said, they use sampling dates as a proxy for infection dates, and I think it needs to be pushed back because probably lineage A was in circulation before, like for some time before it was sampled. And I think in general, why do they even care? Like, was it 2 spillovers? Was it 1 spillover? And I think they care because they're actually trying to explain why is it that the Huanan seafood market is dominated by lineage B, which is phylogenetically a later strain and obviously doesn't fit into the zoonotic hypothesis, because under the zoonosis, if B is descended from lineage A would have spilled over first, so the market should have been dominated by lineage A, as that's a later, that's an earlier genome. But despite the phylogeny, Picard and others like Warby, they're trying to argue that it was lineage B that spilled over first in the market, but then lineage A spilled over second in the market, like a week later. And of course, but even under that scenario, you'd expect to see at least some lineage A in the market, whereas right now the market is almost exclusively lineage B, except for one sample for which I'll talk about in a few slides. And so basically, to make that claim of 2 spillovers work, Picard et al. They have to rely more than just on phylogeny. As I said, phylogeny, B is clearly descended from A, so they have to use sampling dates as proxy for infection dates, and then use those dates to kind of infer when we see a certain lineage introduced into human population. But of course, that approach is in general is potentially flawed, because early data is very scarce and it's anecdotal. When exactly did you have symptom onset? Some people could be mistaken, and especially here, like if we have just 2 people, one with lineage B and one with lineage A, one is saying, okay, I had symptom onset on December 10th, and the other said I had symptom onset on December 15th. As I said, there's an extra mutation for the lineage A person, that could push back the date of the infection earlier. And also, as the market, it was noticed first, like the market was the biggest super spreading location that was noticed in Wuhan, and everyone with a connection to the market was sampled first, because you actually needed a link to the market to get a COVID diagnosis in Wuhan early on. And so as we see that the market is almost exclusively lineage B, so lineage B had a potentially sampling bias, like it's been oversampled in the early cases. Therefore, whatever lineage A genomes, they had a disadvantage, a sampling bias against them, because lineage A was not present in the market and had no patients with a link to the market. And so this bias, it can actually skew the data when inferring spillover date from the sampling date. And that, again, I think I'm beating a dead horse in the Picard model, but by now I think nobody actually thinks there's been two spillovers. Sorry? 45?
30 minutes.
Oh, 30 minutes. Okay. Right. So yeah, basically, in conclusion, I think it's very unlikely that there's been 2 spillover events, which I think by now it's pretty clear. Well, that said, even if there were 2 spillovers, I think it actually would support the lab leak hypothesis better, especially because it's just 2 mutations away between the 2 lineages. And we have examples of lab leaks that, like multiple lab leaks from the same lab, like examples of SARS-1, for example. So it's entirely conceivable for a virus to leak from a lab more than once. But if there really were two zoonotic jumps, we'd expect to find many animals carrying the virus, because for 2 spillovers to occur like in the same location within a week and not anywhere else, I mean that's really suspicious in itself. Like why would there be this kind of exclusivity between the wildlife vendor and this particular market, and only that wildlife vendor would have these infected animals? And anyways, I think especially in light of how SARS-1 happened with like multiple animal spillovers. We had like 5 civet strains of SARS-1 and multiple human introductions, and we have nothing like this with SARS-2. And so it's just very surprising. And like this is 20 years later, we have like much more powerful sequencing technology, and yet just zero evidence of infected animals, infected intermediate hosts. Okay, speaking of SARS-1, when we look back at SARS-1, its emergence, like its pattern, is I think it's like very different from what we observe in SARS-2, because with SARS-1 there's been these like multiple spillovers over several months. Like the virus would get into human population, like try to get a foothold and go extinct, and then but eventually like it actually managed to start spreading human to human. But this is not what we observed with SARS-2. And also, it's not just SARS-1 that's had multiple introductions. Other viruses do the same, like HIV had 4 different spillovers. MERS had like 50, by some estimates, 50 zoonotic introductions, and yet, SARS-2 only had 1. So yeah, basically in summary, if we compare SARS-2 to other outbreaks, SARS-2 stands out as a clear outlier, because normally a virus would jump multiple times from animals to humans and infect them usually at different locations, because there's some sort of reservoir of this virus present, and there's no reason why it wouldn't be able to infect other members of other species at multiple times. But with SARS-2, we have yet to find a single infected animal. And so I think the evidence points to a concentrated human spillover event. And additionally, the A and B lineages of SARS-2 are only separated by 2 mutations, which is unusual for zoonotic spillovers, because typically you see multiple strains with multiple differences between them. Okay, let's now focus on the Huanan seafood market, because I think the entire zoonosis narrative is based on the Huanan seafood market being the location of the animal spillover of the virus. And I think it actually, there's data that the observations we have is more consistent with the Huanan seafood market being just a super spreading or amplifying location of lineage B, because first and foremost, lineage B is a later strain, and market is almost exclusively lineage B. And this is inconsistent with zoonosis, because usually the earliest strain should be dominating the spillover location, like the market. And yet the genomic data of the earliest cases we shows that it was exclusively lineage B in the patients. Like all 16 of the earliest Wuhan patients linked to the seafood market, all of them had lineage B. And yeah, I mean, this is surprising, because lineage A it had 2 mutations closer to that ancestor, but you'd expect, if that was the spillover virus, you'd expect at least some people from the market to have that lineage. But also additionally, the environmental samples also all but 1 were lineage B. All but 1, yeah, and the only lineage A detected in the market from environmental samples wasn't even from the environmental sample. It was from a cultured sample based on an environmental sample, but that environmental sample only had like 22 SARS-2 reads and had a very high CT PCR value, and yet they somehow managed to culture virus, like a viable virus from that sample. But that virus, when they sampled it, not only had lineage A mutations, it also had like 2 or 3 additional mutations that weren't seen until much later in the outbreak. They were like, yeah. So basically all of this is more in line with the contamination, that sample being result of contamination either during culturing or sequencing, and but not like a bona fide virus from, bona fide lineage B virus from the market. And so yeah, basically because of this kind of overwhelming presence of B, like almost exclusive presence of B in the market, I think it's much more in line with the market being just a super spreading location of lineage B or amplifying location of lineage B that got into the market after lineage A spilled over in Wuhan earlier and and basically developed into lineage B, and then lineage B got into the market and got amplified. And the early kind of interplay between lineage A and lineage B in Wuhan is interesting, because even like in the early stages, at least a third of cases in Wuhan were lineage A, and yet we see no lineage A in the market, which is strange. And of course, lineage B had a transmissibility advantage and ultimately outcompeted lineage B into extinction. And so in this case, in this diagram, M is same as lineage B. It's just an old diagram. And all of this is not consistent with zoonosis, but I think it fits very well into a lab leak scenario. And even more, some of the earliest genomes found later during sampling were even closer to the bat ancestor than lineage A. So they had like either at least one more mutation closer to the bat ancestor than lineage A. And so these findings suggest that there might have been an earlier strain that spilled over, like even earlier than lineage A, which then, eventually mutated into lineage B. And one such mutation that's closer to the bat ancestor is this mutation here, C18060T. And several genomes have been found that have this mutation, several early genomes in circulating genomes, and also in some like forensic metagenomic analysis. For example, here this Arctic soil data set that was sampled and sequenced, or sampled in Antarctic but sequenced in Wuhan in like early 2020. They found these SARS-2 genomes from potentially early patients that have this ancestral mutation. And Jesse Bloom analyzed these genomes and reported that there were numerous reads consistent with these being progenitor genomes, suggesting like an earlier SARS-2 in circulation in Wuhan. And of course, Jesse Bloom also is kind of famous for finding these deleted patient data set that had like 11 patients, I think 4 of which had even earlier genomes. Three had one mutation earlier, and one had maybe even 2 mutations earlier, like closer to the ancestral strain than lineage A. And the exact number of mutations is not really important. I think the significant finding is that lineage B was already was not the ancestral lineage, and yet the market is dominated by lineage B, and this is inconsistent with zoonosis, because we would expect, if the market was the location of zoonosis, of spillover, you'd see at least some lineage A in the market, if not like be dominated by lineage A. But and as I promised, there's only one lineage A sample in the market, and out of I think they had 25 or so positive samples, environmental samples. They had 16 patients, all of them lineage B, and then they had 25 environmental samples, and there's only one lineage A from a cultured sample. So the original, and I already mentioned this, the original glove sample had only 22 SARS-2 reads and had a very high CT value for the PCR screening, but yet they managed to isolate a viable virus. And so with additional mutations, like I said, additional mutations not seen in Wuhan at the time until months later. So all of this is really more consistent with contamination during sampling or sequencing. And this keeps... basically, I think I also made this point several times, is essentially the dominance of lineage B in the market is just inconsistent with the market being a spillover of zoonotic lineage. And unlike SARS-1, which had multiple spillovers in multiple locations, multiple animals, with SARS-2 we see none of that. And so this is very surprising, and I think more in line with a lab leak than with zoonosis. Okay, mutation rate. So basically, I think the virus exhibits a very low mutation rate. Like even the early virus exhibits a low mutation rate, in contrast to like SARS-1 or other zoonotic viruses. And this is surprising because you'd expect a virus that spills over to a new host to have a very high mutation rate, especially initially as it adapts to this new host. And here's some diagrams of these like low mutation rate, and I think basically, yeah, they show that from the start it's pretty consistent at like 20 mutations per year. And I think Peter actually challenged this claim and had a figure of 37 mutations per year for early genomes. And so I looked at the paper he cited in this estimate, and it was interesting because he said at the same time that it had 36 mutations in deer, so basically fewer mutations than in humans, but the paper actually had much higher mutation mutation rate in deer, like 1.6 versus 1.3. So if you actually convert it to the full genome at like 30,000 nucleotides, I think you arrive, this should be like 48 to like 52 or something. So not 36, but it's a good paper. I like it because it shows basically compared to alpha and compared to delta, the early mutations in the early strains. And sorry, this is the red is the early strains, the pink is the later strains, and blue is deer. And basically you see deer is still higher, like in their estimate, deer still has higher mutation than the earliest genomes. And the earliest genomes used by that paper come actually from Picard et al. Picard is, everywhere. And Picard actually, their original data, based on which the deer paper used for their analysis, they estimated 27 substitutions, 27 mutations per year based on these slopes rather than 37. So, I mean, again, it's I think the exact number is less critical. What's critical is that even if, I mean, yeah, clearly there's potentially like the first few months of the outbreak, you have a higher mutation rate. But of course, that data is also noisy because Picard et al. They use from like December to February. And the earliest sampling dates, as I said, are very sparse and potentially unreliable because they're anecdotal. But even if you rely on them, the difference between the mutation rate between like early strains and later strains is like twofold between early and later. So basically, they had like 27 versus 20. Like if you take Picard, or 27, like
15 minutes.
Okay. Okay. Right. 15? 15 minutes? Oh, okay. I'm good. Right. So basically, where was I? Right. So the Picard estimate for the early mutation is, for the earliest strains, the mutation rate is 27. The later, like next strain number for mutations is 20, like the lowest is 18 per year. And the deer paper estimated, like it had not the total mutations, but substitutions per site per year. It had like 1.3 for the early genomes and 0.6 for the later genomes. So basically, at most, it's like a twofold difference between the earliest and the later stages of SARS-2 evolution. And at the same time, when you look at what happens when SARS-2 spills over to different animals like deer or minks, its mutation rate gets increased much, much higher. So for example, here it's like between 4 and 13 times higher mutation rate when it's spilled over into minks. And yeah, basically, I think a low mutation rate, I mean, under the lab-leak hypothesis, it's very easily explainable that the virus was already kind of pre-selected for human transmission, but maybe even further adapted. At the same time, I mean, yeah, it could happen naturally as well. Like all of the features that we see in SARS-2 potentially could arise naturally and then spill over naturally. But I think it's in theory at least. But like the number of features that the virus would need to arise, at the same time or very nearly at the same time, I think it's very unlikely for them to arise in a single animal or like single species, especially like definitely not in bats, because as I mentioned, the furin cleavage site or the absence of this N-Glycan, both of them are detrimental to transmission in bats. So it's very kind of surprising for these features to have arisen in SARS-2. But without them, of course, SARS-2 would not be suitable for human transmission, would not be a respiratory virus, would not infect humans. So even despite it having this kind of high affinity to the human ACE2 receptor, without a furin cleavage site, it's still an enteric virus, and without the ablation of this glycan, it would be like 60 times less infectious. So it's just, the number of things that would need to happen at the same time in like between the bat virus and the human transmissible virus are just, to me, a very, how should I put it? Well, surprising, unexpected. And of course, for all of this to emerge naturally, this would probably need to happen close to Wuhan, like emerge naturally in Wuhan, because of course the natural habitat for these viruses and for these bats is like 1,000 miles away from Wuhan, like in southeast Yunnan or like Laos or Vietnam or whatever. And so it's just very odd for this to happen without leaving any trace, like in between, like if it's wildlife smuggling routes, to see no evidence of this spilling over along the way or in some intermediate hosting locations of these wildlife, and yet not see any evidence of a single infected animal, like that's very, very, very surprising. Okay, so I think pretty much done. I mean, this is the final slide, the best explanation slide, and this is the kind of the root claim process where before assigning probability scores, the base factors, basically it's trying to steelman both hypotheses, presenting like the best arguments for each hypothesis and then comparing evidence for both. And I'll just leave this slide to be explained by SAR in session 3, I guess. And this is like a preview of the results, and I think the lab leak probability here it just remains extremely high, of course, based on these best explanations. And I guess we have some time for questions, right? So we have like 8 minutes if I have my timer correctly. If you'd like to ask some questions, finally, I'm surprised that I actually finished before my time. Or we can leave this time for later answering questions, or I'm not sure. But basically, at this point, thank you. If you have any questions, feel free to ask.
Thank you, Yuri. That was very interesting.
I'd like to go back to that kind of my initial question about the kind of what I'm struggling with, or what I'm trying to evaluate, is how much should I think it is suspicious or coincidental that the DEFUSE proposal contains a whole bunch of things that look like genomic features that would be expected, or like that would produce genomic features that we then see in SARS-CoV-2. And so the spirit of my question was, one thing that would help me evaluate that is if there was any sort of data trail or any sort of kind of evidence that any of the DEFUSE proposal specific experiments were being carried out or were being used as a blueprint. So as an example, if you go to your slide 41, Yuri.
Yeah.
Okay, so you're citing this paper here. You've cited the FAL1 restriction site is very interesting, and here in this paper, DNA amplicons were digested using FAL1 with CutSmart buffer purchased from New England Biolabs. Mm-hmm. So this is exactly my question. If this research is done, where, if any, paper trail exists, and if it doesn't exist, why haven't we seen it, given that U.S. vendors are frequently the suppliers of a lot of these things?
Right. Well, I mean, restriction enzymes, they probably have a stock of, them and buying them up years in advance and just would use it. And of course, these days, most of scientific supplies are available in China as well. And so any paper trail you would expect would be in China. And obviously, it wouldn't be released. I don't think they would have used any US or other vendors for their internal like WIV. And they, I think over the years, they also tried to in-house most of the capabilities, like both sequencing capabilities and synthesis capabilities. But even if they need external help, I think they would have used Chinese vendors. There's no need for them to use. And yeah, I mean, synthesizing DNA is these days outsourced to China. Anyways, the most cheapest.
I mean, I think that's a debatable question. I don't. That assertion seems without evidence. More broadly, I buy a bunch of nucleotides from the United States that are synthesized. I think my question is broader than that. Is there any evidence? Like, I understand what you're saying, mm-hmm. that there's a lot of Chinese vendors that can supply this. That makes total sense, and that there wouldn't necessarily be an accessible paper trail. Perhaps even if it was in the United States, this information could be deleted or it could be hard to come by. But so I'm just asking if there's been, kind of any evidence at all of diffuse activity work that was carried out at WIV using this as a blueprint. Like, we have a bunch of speculation, which certainly seems plausible, but is there any other evidence that they did specific things as proposed in WIV? I mean, the one that stuck out to me is like that primer walking. It seems very unlikely that a graduate student, if they were going to primer walk to sequence these viruses, would be doing that synthesis.
I mean, I'm not aware of any paper trail. I'm not sure if anybody looked at the paper trail outside of intelligence agencies. But yet again, my expectation would be that they wouldn't be using, outside providers outside of China. Like, why? There's very, very strong capability in Chinese biotech providers, and so.
Totally. Okay, that's helpful. Thank you.
While we're talking about DEFUSE, I did take some time to briefly look at the DEFUSE grant, and it sounded like a lot of this work was, as proposed by DEFUSE, would have been done in America, specifically in UNC. And the sorts of things that I would expect could lead to a development of a SARS-CoV-2-like virus would not have taken place in WIV as proposed in the grant. Can you account for how in the lab leak theory this virus would have come out of WIV instead of a different lab? Can you explain this discrepancy between the DEFUSE proposal and the lab leak theory?
Yeah, well, I think I mentioned it in the beginning that, DEFUSE was not gospel. WIV could have done whatever they wanted for their own research. DEFUSE is what, EcoHealth wanted to do and get money for EcoHealth. And while in the DEFUSE proposal, the furin cleavage sites were supposed to be done by Ralph Barrick, of course, nobody is prohibiting WIV to later do this kind of work, especially because DEFUSE wasn't funded, and especially it seems that kind of Barrick and WIV parted ways on their furin cleavage sites. But a lot of the work, even within DEFUSE, a lot of the work was supposed to be done by WIV, especially with live viruses. So while Ralph Barrick could have been, this could have been done under DEFUSE is really not a good exercise, because DEFUSE is not limiting WIV to what things they could be doing. They could be, if they have an interest in synthesizing, inserting novel furin cleavage sites, they can do this locally very easily. I mean, they have this capability. And so just because DEFUSE was talking about Ralph Barrick doing it, I wouldn't give any weight to WIV not being able to do this or not wanting to do this, especially if they wanted to do their own stuff for their own funding, internal funding, and not be kind of beholden to NIH or EcoHealth or whatever.
Just to be clear, do you have a reason to say that Ralph Baric stopped collaborating with people other than the fact that they weren't co-authors on a specific paper? Or I mean, obviously, because I don't know the people involved, it's very hard for me to have a feeling for who is working with whom, why, that sort of stuff.
Right. No. Basically, the paper, not paper trail, but the paper is published by Baric in like 2019 and afterwards. Especially this particular paper that is precisely about the things that they collaborated with WIV in the past several times, both DEFUSE proposal and 2015 they done this very similar work together. All of a sudden they're starting to do this separately. So, I mean, to me, this is a signal that they're no longer collaborating on these types of things. So, I mean, I haven't interviewed Ralph Baric to ask him like, when did you stop collaborating with WIV? But this could be a good question to ask him. Maybe at some point he'll provide an answer.
Maybe going off, I want to. I'll have more questions about this during rebuttals, but back to FAO1 and CCCG. So one of the reasons, like, it's a very interesting idea that this introduces a novel cleavage site that could be used in analyses, and that I think is very interesting. One question I have about that is, what is the scale of viral engineering going on? And the reason I ask that is I could understand like a RIFLIP analysis, or just basically like a simple gel analysis to say, okay, our culture has lost the furan cleavage site. But if you're doing this at a larger scale, if you've got 1,000 cultures going, or you've done 1,500 or something like that, it seems unlikely that you do that versus sequencing, especially given the context that you said that they have high levels of sequence capability. So I want you to speculate or kind of compare in what sequences that FAO1 cleavage site would be valuable. What number of viruses would they have to be engineering for that to be the preferred method versus using their in-house Illumina?
It's a good question, and I think for me, sequencing they have to like pass along to some other group and have to wait, and I mean, if you're sequencing, could also be a cost issue as well. Whereas if you can run a simple gel with a simple digest with like primers that you already have that can amplify this region and like quickly screen your colonies, if you're doing a lot of experiments and you want to do them quickly, and not have to pause every time for sequencing at each step, I think this could be useful. But of course, sequencing, like this is for like quick screening, and sequencing can be the validating step to make sure that you're not having some sort of false positive. In terms of like the number of viral culture where this becomes kind of intangible or not feasible, I'm not sure. And they could have like this, if they're using multiple viruses for screening multiple viruses, and if they're engineering novel furin cleavage sites in multiple viruses, they could have had the same furin cleavage site in all of these viruses with the same kind of tracking beacon that they could quickly do analysis like on a panel. Okay, which viruses had this furin cleavage site get deleted, in which culture conditions, or which, like, they could have then iterated on the actual furin cleavage sites and have different restriction enzymes based on like the underlying nucleotides. Maybe they wouldn't have like RRAR and is one example, but then they would have K or RARR, and they would use a different restriction enzyme that's like for those nucleotides that would work with the RA, and then they would have multiple screens going in parallel, and it's just very easy to run a bunch of gels for this screening and quickly have your result rather than have to wait for sequencing. But I mean, this is all speculation, so I mean, it's as good as my guess is as good as yours.
No, very useful. Okay, well, I think let's take a 5-minute break, and then Peter, do you want to go after 5 minutes? Okay. All right, everybody will return after 5 minutes. I'll leave the video running this time, and then I will stop it after Peter's speech to give our mid-break.
Thank you, Yuri.
Okay, so I stopped sharing. Is that good?
Yep, great.
All right.